Dear Philip, dear colleagues, first of all I would like to thank you for inviting me and to show our results of the last years. I'm head of a laboratory virus safety of xenotransplantation at the Institute of Virology of the Free University in Berlin. And this laboratory is part of a German-wide Transregio consortium, which is working in the field of xenotransplantation. There are groups generating multiply genetically transformed pigs. There are groups transplanting pig islets, pig kidneys and pig hearts to non-human primates, and there are immunologists and virologists. As Hervé and you already said, the main fact in understanding of retroviruses is that they are able to transform their RNA into DNA, and that they can integrate the DNA in the genome of the cell. And in the case of HIV, this happens in the infected lymphocytes, but never in liver cells. But in the case retroviruses hits an oocyte or a sperm cell, then this integrated virus is in the cell of the new organism, and so it's in all cells of the pig or of the human or of the mice, and this we call endogenous retroviruses. Another important feature of retrovirus is that their transmission from one species to another. It's well known for HIV-1. They are the result of transmission of immunodeficiency viruses from different species. They are non-pathogenic in the natural host, but they are induced severe immunosuppression in the infected host. Such a species transmission is also described for the koala retrovirus, and as I will show later, for the porcine endogenous retrovirus, there are the roots of these viruses in different rodents and other animals. And this is a schematic presentation. You already have seen it. There are on both sides of the integrated provirus the LTRs. There is a GAC polymerase, the protease, the envelope protein, and different sub-units, for example, the integrase. And what happens when a retrovirus integrates? And this can lead to a mutagenesis. If it integrates in an inert region, in intron, for example, there is no regulation, but it can be used for, there's no expression, but it can be used for the regulation of other cellular genes. And if it integrates in an active region, in exon, then there may be expression. But especially the human endogenous retrovirus are well known for the fact that they are rapidly silenced, either by DNA methylation or excision. So here is an example how an integrated LTR can influence the expression of a cellular gene by transcription factors or by enhancers from the LTRs. So they can really regulate the expression of cellular genes. And the question is, why do we have these viruses in our genome? Is there no possibility to kick them out? And the answer is easy. There's no way to remove them, because we don't have a mechanism to remove simply pieces of our genome. The only possibility to lower the proviral load is that there is a recombination between both LTRs. Then the provirus disappears, and we have solo LTRs. And for example, in the case of HERV-K, we have 600 copies, and there we have more solo LTR as intact viruses. And here are a lot of examples of the influence of viral integrations of different elements on normal cellular genes. And the most important I only will mention is the HERV-E. It came into the ciliary gland and regulates the expression of this amylase gene, and this leads to the fact that we can digest bread already here and not only in other parts of the body. What are the consequences on the protein level? First, the reverse transcriptase is also able to transcribe, let's say, messenger RNA into DNA, and then this DNA can be integrated as a pseudogene of a cellular gene. Then we have core protein and surface envelope protein. They can protect from infection of other retroviruses, either because they are on the surface and hinder the infection, or like GAG, they are intracellular restriction factors which can fight with other retroviruses and not only with retroviruses. And we have the transmembrane envelope protein of retrovirus, and they have two functions I will explain in detail later. The fusion peptide is important for the syncytial trophoblast and in the placenta, and it is also important for immunosuppression, because all retroviruses, not only HIV-1, are immunosuppressive viruses. Here is HERV-K, which we studied in more detail. It is a more complex virus. It has additional genes in addition to GAG, POL, and PRO and ENV. There is REC. It is an equivalent to the REF of HIV-1, and there are HERV-K forms in the genome which have a deletion in this region, and they have another additional protein, NP9. And you can imagine that there are full-length and spliced messenger RNA, and based on these different sequences, different messenger RNA expressed, we developed a system where we have primers detecting full length and spliced from the accessory proteins, and then we see such pictures, for example, in a melanoma cell line or in a teratocarcinoma cell line. So we see full-length and spliced messenger RNA, and this is not the case in immortalized human embryonic kidney cells. So we see that there is a very strong expression not only of the full-length but also of the spliced messenger RNA. And doing this for numerous tissues, we found that, for example, the REC I mentioned is expressed in testicular tumors. The ENV is also expressed in some tumors or normal tissues, this is also expressed in some, but in normal tissue, the full-length messenger RNA is always expressed, but not the spliced. Here, for example, you see a protein expression having an antibody against the transmembrane envelope protein. Here we have the teratocarcinoma cell line, here we have the melanoma cell line, and here we have immortalized kidney cells. And in the case of the teratocarcinoma cell line, we even can see virus particles. More important is the search of antibodies against HERV-K. When we screened, for example, testicular tumor patients, we had a very high percentage of these patients being positive tested. And it was also interesting, after treatment of the tumor, the antibodies disappeared. Similar results we have for some lymphoma patients, and most interesting, we had also antibodies in HIV positive patients, but this is obviously due to the cross-reactivity between both viruses. And in pregnant women, we had also a high number of positive women, especially when they had more than one children. Here again, the REC, which has several functional domains. It mediates the nuclear cytoplasmic translocation of the RNA. It has a nuclear location sequence and an export sequence and an leucine-rich domain, and it may interact with some sequences and proteins which may support cell transformation. But in other cases, I guess that the expression of endogenous retrovirus is simply the result of the extensive proliferation of tumor cell and has no effect, and it's not the cause of the tumor development. The porcine endogenous retrovirus are also found in melanomas. Here are pigs with a melanoma on the back and metastasis in the liver. We found expression of spliced and full-length messenger RNA of different pervs, and we even found virus particles in cell lines which were derived from this tumor. The most interesting fact of the importance of endogenous retrovirus is their role in the placenta. I mean, we wouldn't sit here without our endogenous retroviruses, and they are expressed in the placenta, and they produce at the edge of the of these parts of the placenta. They are using their fusion properties. They from the trophoblast cells, they produce a syncytial trophoblast, which is responsible for nutrition and gas exchange. And it was nicely simulated in culture where you have cells expressing this transmembrane and surface envelope protein. The surface envelope protein interacts with the receptor on the neighbor cell, and then the transmembrane envelope protein induces the fusion, and you have a syncytia. And this is the same process which takes place when a retrovirus infects. The retrovirus has the surface envelope protein and the transmembrane envelope protein on the surface. It interacts with the receptor on the cell membrane. Then the fusion peptide of the transmembrane envelope protein jumps out into the cellular membrane, and then there is a fusion between cellular and viral membrane, and the virus can enter the cell. And here this process is also simulated. Syncytin is not only present in primates. We have syncytin 1 and 2, but in different other species. And it is important that to note that there are different types of placenta documented here, and in all these cases you have fusion of cells of both parts, and this is due to the presence of the syncytin in all these species. Interestingly, in pigs, they have a different placenta. At the moment, nobody was searching for syncytin, and the question is open whether they have or whether they need or not. And here is a full table of from one of my reviews, where all these syncytins in all species are shown, and the different types of placenta are shown. And for example, when they integrated in human, we have syncytin 1, which integrated 25 million years ago, and syncytin 2, which was quite earlier. And in cows, for example, it is well studied that first one syncytin fulfilled the task of fusion, and later another virus infected, and the other transmembrane envelope of this new virus took over this function. So they are even changing the viruses which are important for the placenta production. And we, of course, we wanted to know whether HERV-K is also present in addition to syncytin 1, which is HERV-W, and syncytin 2, which is HERV-FRD. So we were looking for HERV-K, and we found HERV-K not in the syncytial trophoblast. Here, the outer lane, this is the syncytial trophoblast, but we found HERV-K expression in the next layer of cells. So we showed that it is also expressed. So we have at least 3 endogenous retroviruses expressed in the placenta. Here we showed it in Wester plot with mouse antibodies and goat antibodies. So you see that with time the expression increases. Here are the controls. So it is present, and the question is, what does HERV-K in addition to syncytin 1 and 2? We also found expression of HERV-K in human stem cells. Full length and REC NP9, all full length and spliced were expressed in a stem cell line, and when they differentiated this line into the neuronal direction, the expression decreased in 2 lines shown here, and you also can see here expression in the stem cell line, and then after differentiation the expression disappeared. Here is the control, the teratocarcinoma cell line. So it is important, especially for stem cell therapy, because if you give the stem cells which are not differentiated, it happens what was described in similar experiments in mice. You get a teratocarcinoma in the recipient. There are a lot of people saying, well, if you have expression of the endogenous retrovirus on the surface of the tumor, you can use them as a target for immunotherapy. And I was discussing this in numerous letters to such publications, denying that at the moment we cannot use them, because first they play a role in the placenta development, but I mean, if you have a tumor you don't need a baby, so this is not so important. But as we showed, it is expressed on human embryonic cells, and this may be dangerous because they may be in different organs And we also have shown in mice and in pigs that they are expressed in immune cells, and so this may be also dangerous for immune cells if you treat a cancer patient with antibodies against an endogenous retrovirus. As I said, retroviruses are immunosuppressive, and my laboratory has shown that disrupted retroviruses purified transmembrane enveloped protein and peptides corresponding to a highly conserved domain called immunosuppressive region in the transmembrane enveloped protein. They inhibit mitogen-driven lymphocyte proliferation. They modulate cytokine expression. For example, IL-10, IL-6 is upregulated. And we have shown that single mutations in this immunosuppressive domain abrogates this immunosuppressive activity, and this was shown for HIV-1, for HERV-K, for the koala retrovirus, and for the porcine endogenous retrovirus, and I think this is true for all retroviruses. And here you see a comparison of this immunosuppressive domain, which is located in, here you have ENV, surface envelope protein transmembrane, and here you have the immunosuppressive domain, and you see it is highly conserved. Here you have PERV, feline leukemia virus, mouse leukemia virus. Then you have syncytin 1, which is HERV, syncytin 2, HERV-FRD. You have it in the syncytin of mice, cow, sheep, and so on. You have it in other endogenous retroviruses of the humans, and you even have it, and this was the reason why I published this, you have it even in the placenta of a viviparous lizard. And you have it in HIV-1, and you have it in mouse tumor virus, and you have it in HERV-K. And let me come quickly to the porcine endogenous retroviruses. These are major viruses and budding viruses. We have 3 types in the genome of the pig. PERV A and B is present in all pigs, C in most. They are released from normal pig cells, and they can infect human cells, and therefore they pose a risk of xenotransplantation using tissue cells or organs from pigs. And the xenotransplantation has great achievements in the last time. Islet cells survive in non-human primates for 950 days, heterotopic heart 945, autotopic heart 195 days. This is a record held by our consortium in Munich. Kidneys, neurons, only lung and liver are not so long, and you certainly heard that in January of this year the first transplantation of a pig heart into a human patient was performed, and the patient survived 2 months with this pig heart, and this is an enormous success because the first allotransplantation by Christian Barnard in 1967 lived 18 days, and the first allotransplantation in Germany only 27 hours. So the, 2 months is a success, and unfortunately the colleagues in the United States in Baltimore, they transmitted a porcine virus, PCMV, to the patient, and this certainly contributed to the early death. So as I said, we have PERV A, B, and C. They can recombine, and these PERV AC, they after passaging, they are high titer, and after passaging on human cells, they acquire regulatory sequences in the LTR, and this increases their titer again. As I already said, these are the origin of PERV A, B, and C, and they have also different LTRs. And we determined the number of the proviruses using droplet digital PCR. It's around 60, also in normal commercial pigs. And I have to add that they are still active. So when you measure the copy number in different organs, you get different results. So this means the virus still can replicate in the living pig and increases the number of integrated proviruses. How to prevent such a perf transmission? My laboratory was working a long time on this. First, you have to select perf-free pigs in order to avoid this recombination between A and C. Then we studied siRNA, and in vitro and in vivo we have had pig which expressed siRNA, and these pigs had a very low level of expression. And we have vaccines against porcine endogenous retroviruses, which are neutralizing and which can be used. And we were the first to study gene editing. We used the zinc finger nuclease. Unfortunately, the zinc finger nuclease was toxic, but an American Chinese group, they used CRISPR-Cas, and they were taking embryonic fibroblasts using CRISPR-Cas, inactivated all integrated perfs using CRISPR-Cas specific for a highly conserved region in the polymerase. And then by somatic cell transfer, they put it in an oocyte, in a surrogate mother, and they have newborn pigs with inactivated perf. And we have problem with this because in all preclinical studies no perf was transmitted, and in the clinical studies, for example in islet cell transplantation, in order to cure diabetes, also no perf was transmitted to human recipients. So the question is whether it is necessary, because you have off-target effects of CRISPR-Cas, and so this is an open discussion, but it is theoretically possible. And finally, I would thank my colleagues at the Paul-Ehrlich Institute, the Robert Koch Institute, and now at the Institute of Virology, all our national and international cooperation partners. And I would like to thank you for the invitation and for the attention.